Hello and welcome to Chemspec Digital 2021. My name is Imran Omar and I have the pleasure to introduce the next presentation in the RSC Lecture Theatre. But before I do, please note that we are recording this presentation to make it available to view on demand. Attendees are invited to write questions into the chat function from where we will read them out loud at the end. And now I will hand over to Dr. Megan Smith of Almac Group, who will be presenting a contract development and manufacturing organization strategy towards the implementation of continuous flow. Over to you, Dr. Megan.
Okay, thank you, Imran. So hello, everyone. Thank you for taking the time to join me today. As Imran has said, my name is Megan Smith and I am technical leader within our custom and flow chemistry group here at Almac Sciences. So I've been with the organisation now for just over 5 years, initially joining as a biocatalysis chemist before moving up through to my current role, where I am responsible for the development of continuous flow chemistry and their associated processes within our technology group. So as Imran said, my talk today is titled A CDMO Strategy Towards the Implementation of Continuous Flow. And I will give you a brief overview as an introduction to what our strategy is here at Almac, and then go into some technical case studies as well. So see if we can get to the next slide. There we go. Okay, all these platforms are always different. So normally, whenever we give this talk in person rather than virtually, one of the questions that we'll ask the audience is, what's the first thing that you think of when you hear Northern Ireland? And quite often we get responses such as the Giant's Causeway, more recently as well, for Game of Thrones has become very popular, or some of the car fanatics out there might suggest the DeLorean. But one thing that might be a little bit more unexpected would be the Buckminster fullerene. And you might think, what has that got to do with Northern Ireland? But we believe that we might have the world's largest Buckminster fullerene. So this is on the main road as you come from Dublin to Belfast, and it's an art sculpture, which of course was not meant to be representative of a Buckminster fullerene, but for us scientists, we can't help but get excited about these things. So hopefully after the pandemic, if people have the chance to visit Northern Ireland, they might also consider having a look at what is affectionately known as the Balls on the Falls, due to the local road that it's positioned on. But with that, then I'll introduce the Almac Group for anyone who might not be aware of us and what we do. So we are headquartered in Craigavon, which is about a 30-minute drive from Belfast, Northern Ireland. And we employ over 5,000 people globally, and just recently celebrated our 50th year. So the Almac Group provides an extensive range of services for the pharmaceutical, biotech, flavour and fragrance, agrochemical industries globally. And these services range from, as you can see at the bottom here, R&D, biomarker, discovery, development, commercialization, API manufacture, analytical testing, right through to commercial scale manufacture. And clients can use a number of integrated services across the development pipeline, or select standalone services to suit their requirements. So I am part of our Almac Sciences division, and more specifically work within our technology group, which we offer a range of services from biocatalysis, which I'll touch on today, isotope labeling, physical sciences, and of course, flow chemistry, which will be the main part of my talk today. So some of the audience at Chemspec may be more familiar with Iron Chemical Company, which is a member of the Almac Group since 2016. So in Iron Chemical Company, we have a wide range of expertise from biocatalysis, chemocatalysis and ligands, as well as making aroma products for the flavour and fragrance industries, and optical monomers as well. So if anyone in the audience today has contact lenses in, quite possibly you have a little bit of Iron Chemical Company with you and you didn't even realise it. My colleagues are exhibiting, so if anyone has any specific queries, please don't hesitate to get in touch with either me or with them directly. So Most of the audience are probably fairly well versed in the advantages of continuous flow. And really for us here at Almac Sciences, the purpose of our flow chemistry platform is to be able to provide superior solutions for our customers, utilising the best available technology. And really a key driver for us is to unlock reaction classes which are inherently more difficult to scale in batch mode. And really our focus is on identifying opportunities for our customers where we can save on processing time, costs, or access new synthetic strategies towards complex targets that may not have been previously achievable using batch processing. So in terms of our strategy then, on this slide, you can see an Ansoff matrix with the 4 quadrants detailing our strategy for implementation of continuous flow for existing and new markets, as well as existing and new products. So in terms of market penetration, the COVID pandemic has really highlighted for everybody within the chemical sectors globally the importance of security of supply. And we have a number of products that we've been looking at where we're considering the use of continuous flow to reshore key building blocks, and also for targets which are increasing in quantity, we're looking at flow as a way of unlocking additional capacity. So initially then, as we started to look at continuous flow here at Almac Sciences, one of the key areas that we wanted to look at was unlocking those reaction classes that we can't access in batch typically. So for us, we looked at high energy transformations, which I'm going to give a case study on today. We also had looked at hydrogenations as well, and oxidations and photochemical transformations. As well as I've said, we work within our technology group very closely with our colleagues in biocatalysis. And that has allowed us to demonstrate the synergies between the 2 technologies. And I'm going to present to you today where we've used enzymes as a purification technique. And this is really allowing and enhancing our market penetration. As well, we've been doing a lot of work on the area of cannabinoids and have built up extensive expertise in this. And of course, then we have diversification considerations to maintain a competitive advantage. And for us, this is really through our R&D collaborations, and they allow us to undertake what would be considered to be higher risk projects, but these are also very high reward. So we have a number of ongoing academic collaborations through which we've published 8 peer-reviewed publications since 2020. So we have 3 PhDs in collaboration with Queen's University under Horizon 2020 funding, a project called AppFlow. We also have a KTP, which is a knowledge transfer partnership with Queen's University Belfast, and we have a number of ongoing collaborations with University College Dublin, including a Science Foundation Ireland Fellowship, as well as a number of PhDs. So that really is putting into perspective our strategy within Almac in terms of flow chemistry. And recently we have had a £5 million expansion for an R&D centre, which is home to our Department of Technology. So that's where I'm sitting at today here in Craigavon. And you can see through some of these photographs, some of our capabilities and kit that we have, including high pressure hydrogenation, which I'll talk about in more detail. As well as we've invested heavily as well in the Chemtrix technology, as it gives us a seamless scale up from lab scale proof of concept through to low kilo manufacture. So hopefully that has put into perspective a little bit about our strategy, and now I'm going to go through some of our case studies that we've been building in terms of our expertise. So one of the key areas where we wanted to develop flow capabilities was for hydrogenations, and within our GMP plant here in Craigavon we have the capacity to do up to 1,000 litres and up to 5 bar pressure. But within Iron Chemical, which is our non-GMP manufacturing facility, we currently don't have any high pressure hydrogenation capabilities. And the use of continuous flow would give us access to much higher pressures and throughputs as well. So as we began to think about performing hydrogenation reactions under flow, one of the first interests that we had was in nitro reductions. So approximately 65% of drugs on the market contain an amine building block, and as a result, new synthetic methodologies to access them are always in high demand. So in our work, we started by considering 3 aromatic nitrobenzoic acids, which when hydrogenated would give us access to 3 synthetically useful amines. So namely PABA, so para-aminobenzoic acid, which is often taken orally to treat skin conditions. MABA, which is a precursor to methyl anthralenate, which is a grape flavoring. And then anthracenic acid, from which indigo dye can be obtained. So in the development work for this project, what we found was that with the presence of the carboxylic acid functionality, we could exploit that and as a result run the hydrogenation in purely aqueous alkaline conditions. So flow hydrogenation offered several advantages over batch, including the increased safety due to the smaller reaction volumes and improved outcomes as a result of increased substrate to gas to catalyst interactions. As well, often flow will give you reduced side products due to the faster reactions that can be achieved. And critically, and one of the big drivers specifically is the ease of downstream processing, as you don't have to separate the catalyst from your reaction in the workup. So we have focused in Almac on packed bed reactors, which are simple to handle and easy to operate. And we have a range of pelletized catalysts available for rapid screening to demonstrate proof of concept. So on this slide, you can see 2 of our flow hydrogenation development rigs. So on the left-hand side is a custom built system. And to give you an idea of scale, this fits within a walk-in fume hood. And we've also recently investigated in the commercially available Hellcat system, and really the driver behind this was to access the higher pressures and temperatures that our customer projects were demanding. Our custom built rig then affords us the opportunity to have a direct scale down of a system which we are currently in the process of designing and commissioning, and which should be capable of 100 kilos a week throughput dependent on the chemistry, and that's to be sited within our new GMP expansion here in Craigavon. So as with any flow process that we develop here in Almac, the first port of call is typically the batch hydrogenation. So this really allows us to get an indication of solubility as well as of the necessary temperatures and pressures that are required for the reaction under investigation. So in this work, we investigated catalyst loading using palladium on carbon, as you can see in the graph on the left, which unsurprisingly gave us the indication that higher catalyst loading would result in faster reaction time. And we also investigated the effect of hydrogen pressure on the reaction. And as you can see here on the right-hand side graph, that 10 bar hydrogen pressure gave us a faster reaction than 5 bar. And one thing that we critically noted here was that 100% consumption of the starting material did not correlate to 100% of the product aniline. So this was most likely the formation of genotoxic impurities as intermediates. So as we move to our packed bed reactor to investigate the flow hydrogenation, the first series of experiments that we did indicated that the reaction was proceeding much quicker than in batch mode. So with a 2-minute residence time, we were getting quantitative conversion, but for the corresponding batch reaction, if we sampled that after 4 minutes, we were seeing less than 20% of the product. What we also noted in this instance was that the effect of the hydrogen pressure was much less pronounced. So it was only marginally faster with 10 bar pressure compared to 5 bar pressure. So under typical reaction conditions, a mixture of the reactive gas and liquid that flows through the packed bed reactor assumes a trickle flow behavior, which gives us a thin film of liquid that's in contact with our solid catalyst particles and in tandem flow with that gas layer. And it's really this configuration which permits rapid diffusion of the liquid, which gives us very fast mixing of the dissolved gas, substrate and catalyst, and consequently results in that faster reaction rate. So one thing that was very important was to demonstrate our efficiency of our packed bed over time. So we were able to run this process for 6 hours, getting greater than 95% conversion to the desired amine. And there's absolutely no evidence of any of the GTI impurities in the flow process, which was most likely due to the greater availability of gas in the flow regime. We were able to run this process for a number of days consecutively over a week, which showed the packed bed was tolerating the reaction conditions well. So there were a number of benefits to the flow process over the batch, including both higher productivity and space-time yield. The potential for less capital outlay due to being able to reuse the catalyst, and also easier downstream processing, as we did not need to filter to recover the catalyst, which typically also results in better purity as well as safety. In this case, the flow process was environmentally favourable, as we were able to run it in purely aqueous compared with the organic batch process. And critically, we were able to demonstrate the robustness across ortho, meta and para derivatives, as well as show that increased control over the GTI impurities. So following on from this work, we were able to identify an opportunity in a customer project where we could exploit our knowledge. So this was a process that was being run in Iron Chemical Company as a batch transfer hydrogenation. And it suffered from a very difficult product isolation due to the workup. So under our standard reaction flow for flow reaction development, we were able to perform rapid screening of our library of pelletized catalysts to go through process optimization and ultimately deliver a kilo of material for downstream processing assessment. And the flow process removed the need for organic solvents and formates from the process and eased that product purification as well. And critically as well then offered us the increased control over those GTI byproduct intermediates. So flow hydrogenations are a critical strategic target for us here at Almac Sciences, and we have a number of live projects ongoing, including nitrile reductions, as well as one for saturation of an aromatic ring. So moving on then from hydrogenations, high energy transformations are also a very important part of our flow strategy here at Almac. So high energy transformations and reactions involving gaseous species are often exploited in continuous flow for industrial applications. And this can really be rationalized by the multitude of challenges and risks associated with performing these on scale in batch. And often, sometimes people refer to these as forbidden reactions or forbidden chemistry. And really, as most people will know, it's the miniaturization of those flow reactor components that enables the effective heat and mass transfer, and critically limits the amount of hazardous reagent in the reactor at any one time. So Curtius rearrangements are invaluable for converting carboxylic acids to amine derivatives, however, relies on the formation of acyl azide intermediates that release nitrogen gas upon heating. And in this workflow also offers an effective telescope means for derivatizing the potentially unstable isocyanate intermediates towards, in our case, we chose to go to carbamate products. DPPA is chosen because it allows direct activation of the carboxylic acids, but also critically because of its stability and commercial availability, as well as offering a homogeneous liquid phase system. So a major hurdle, as any flow chemist will know, is reactor fouling due to precipitation. And typically this is caused either by the low solubility of reagents or by precipitation of byproducts formed in the reaction. So during our development work, we were able to synthesize a library of products with the NCBZ moiety, which were generated from a Curtis rearrangement using benzyl alcohol as the nucleophile. And as with many of our flow processes that we develop, solvent choice proved to be a critical parameter to consider. And when this was being run in acetonitrile, trace amounts of water resulted in the formation of an insoluble urea side product, which caused reactor fouling. But when we switched to toluene with its higher boiling point and lower propensity to contain significant amounts of water, this was found to be a very good choice for this chemistry. So if we consider the schematic here on this slide, we have flow process with two feed lines that mix at a T-piece before passing through our heated flow coil. So in our first feed line, we have our substrate, which was sparingly soluble in toluene, but with the addition of triethylamine, the corresponding salts were soluble at a concentration of 1 molar. And also in this first feed line, we have our nucleophile, so we have our benzyl alcohol. In our second feed line, then we have our DPPA in toluene, and we actually limit the stoichiometry of this to 0.9 equivalents to prevent contamination of the product stream with residual azide species. So in our initial development work, we determined that we needed a temperature of 120 degrees Celsius and a 30-minute residence time to yield full conversion of our substrate. We did observe a biphasic slug flow regime due to the formation of the nitrogen gas. One other thing that was important was that we found that we needed a slight excess, so we needed 1.8 equivalents of benzyl alcohol in order to achieve high isolated yields. So we had successfully demonstrated that we could do the Curtis under flow. However, because of that excess of nucleophile, which many Curtis rearrangements require, we now had a product stream which was contaminated with residual benzyl alcohol. And the boiling point of benzyl alcohol is very high, it's 205 degrees Celsius. So we began to wonder, as I said earlier, we work very closely with our colleagues in process chemistry and biocatalysis. We started to wonder which options from within our science toolbox could we exploit to help solve this problem. And that really led us to consider our traditional process chemistry options. So we thought about evaporation, extraction, chromatography, or crystallization. Or we wondered if we could exploit our biocatalysis expertise for purification. And this is when it started to get really exciting, because we realized that if we used CalB, which is a hydrolase enzyme in the immobilized form, that we could demonstrate the synergies between both our biocatalysis and flow chemistry platforms. So what we realized is that we could exploit the chemoselectivity of enzymes to effectively tag and modify the residual benzyl alcohol. So using CalB, we were able to convert it to benzyl butyrate in the presence of vinyl butyrate to isolate the desired carbamate products. So we modified our initial flow setup, as you can see here, to include a third stream of vinyl butyrate in toluene, and we needed 3 equivalents of this. So the stream of vinyl butyrate and toluene mixes with the crude reaction stream that's coming out after the Curtis rearrangement before passing through an omnifit glass column, which is packed with CalB and run at ambient reaction temperatures. We did see some discoloration, as you can see in the photographs of the CalB columns. However, the enzyme is particularly robust, and this was proven to be inconsequential to the enzyme activity. So as you can see here then on this slide, we were able to demonstrate the effectiveness of the immobilized CalB to catalyze the conversion to benzyl butyrate with no benzyl alcohol observed after 3.5 runtime, as our runtime is shown on the proton stack plot. The pure carbamate product could then be isolated following workup and crystallization from heptanes. So the associated safety benefits with flow for handling of hazardous or highly reactive intermediates are often exploited to offer industrially relevant and scalable Curtis rearrangements. However, with the required excess of nucleophile, often it leads to complications in your workup procedure. But what we've shown here at Almac Sciences is that we have been able to develop a facile purification process under continuous flow. So then we began to wonder if we could exploit the this continuous flow approach further by converting the selected carbamate products into derivatives of beta amino acids. And we really had two options that we were considering. We had conventional alkylation of the carbamate with a suitable electrophile, but it had a number of drawbacks, including the need for a strong base and per atom economy. But with option 2, which is the one that we chose to do, we realized that we could utilize electron pair alkenes as reaction partners to undergo an azo-Michael addition reaction on our CBZ carbamates. So we used two methods to trial this. We used initially potassium carbonate as a heterogeneous catalyst and found that we needed very short residence time, so 5 minutes at 100 degrees to achieve high conversions. But we were concerned about the scalability of this approach, so we looked at a second approach using a biphasic liquid-liquid system with sodium hydroxide, where we saw a biphasic slug flow pattern, and we were able to obtain the products in high yields after phase separation. So then we looked at the methyl ester substrates that we had synthesized and found that we could perform the alkaline hydrolysis of the ester group, and it required quite a long reaction time. But importantly, we were able to achieve this without cleaving the carbamate group, which allowed us to have further and protected amino acid species that we could use further. And just a final example of some of the Curtis work that we've been doing, where we wanted to expand on that integrated enzymatic impurity tagging strategy that we had developed. We looked at the use of Bock protected proline species, which had substitution on the chiral carbon. So we anticipated that we would get the isocyanate intermediate 5, which you can see on the slide here, to afford versatile alpha aminopyrrolidine species, which could be potentially useful amine building blocks. However, we didn't actually get the CBZ carbamate, but the ring opened ketone, which gave us access, which we were quite excited about, to 1,4-di substituted products, which aren't necessarily the easiest products to access, typically having to go through a Stetter reaction. So just as an aside at the end here, we've been doing some work through our collaborations on benzylic oxidations using light, and we've been using some of our microbubble technology that we have in-house here. So we have a microbubble generator, which was initially actually developed in Japan, where they wanted to supersaturate the water where they were growing the fish for sushi to see if they could get higher value by getting bigger fish. But we've been able to use this in a chemistry setting, and we definitely don't have any fish tanks in Almac Sciences here. And what you can see from this graph is that we were comparing for our benzylic oxidation the use of compressed air with pure oxygen versus microbubbled air. So with compressed air, we were getting low conversions, about 10%. When we increased to oxygen, we had about 30% conversion, but we were able to use air much more effectively than pure oxygen using our microbubble generator. And we're hoping to publish this. We've got a manuscript in the final stages of preparation, and we're hoping to publish this very soon. So maybe next year at Chemspec in person, I'll be able to tell a little bit more of this story. So that there, in summary, really just highlights some of our strategy here at Almac Sciences. Of course, in the interest of time, I haven't been able to touch on everything. We've been doing some in situ bromine generation as well for some of our key building blocks that we're trying to reshore. And just finally, before I finish, just to acknowledge the team here at Almac Sciences, who have obviously put in a lot of the work, as well as our academic collaborations, so through UCD and Queen's as well. So with that, I can hopefully finish on time and hand back over to Imran to let him see if we have any questions for our Q&A.
Thank you so much for the presentation, Dr. Smith. We're now answering some questions that you may have. And we have a first question here, which is basically, let me just see what's going on here. All right. Is it the gas liquid separator obligatory? Moreover, how do you manage the excess of H2 or safety issues?
Okay, so yeah, so the systems that we have are in fume hoods, so we're able to vent that. So we have an excess of hydrogen there. We use a cyclone, so we have the gas and liquid coming in, and the gas gets vented off the top. And then we can collect our liquid, which is obviously our product solution. So in terms of the hydrogen, we have all of the necessary safety in place here at Almac. We have hydrogen detectors and automatic hydrogen cutoffs as well, but typically it gets vented out through the fume hood at this stage. For larger scale manufacture, that will be an engineering consideration for us here at Almac.
And we have a second question. How scalable is your enzyme purification stage strategy?
Yeah, so in terms of the enzyme purification strategy, so that enzyme is immobilized, it's commercially available. And in terms of scaling that up, you obviously just need a slightly larger column. It's similar to the hydrogenations, where you would consider your catalyst size as well in terms of aspect ratio within the column. Here at Almac, we have extensive experience in enzyme immobilization, as well as suspension polymerization. And we had a recent project that we were working on in flow, where we were actually making polymeric beads. So we were able to make those of various sizes, so larger sizes, smaller sizes, depending on the droplet size that we were forming. So there's definitely, in terms of Almac, being able to scale that up, we can certainly look at all aspects of that, from the column size to the actual formation of the catalyst particles as well. So it's certainly scalable.
Okay. And we have one last question, which is, how does a typical project run within Almac Sciences?
Okay, so yeah, within Almac Sciences, all of our projects for flow typically follow a 4-stage workflow. So we would have initial proof of concept, where we will either tech transfer a process that our customer has maybe supplied and demonstrate it in our assets, or do proof of concept. So typically we would get a batch process or procedure from a customer, and then we would look at and evaluate that in flow. So really, initial proof of concept is about demonstrating reaction validity in flow. And once we have that, we would move on to stage 2, which would really be our process optimization. And that's really where we're wanting to find out about the limits of the process. So where will it fail, what's the maximum temperature, the minimum temperature, etc., and really test the robustness of that process. And then typically what we do next in stage 3 is we deliver a low kilo quantity of material, so really demonstrate it on pilot scale. And then further to that, the project would move towards larger scale manufacture.
Okay, well, I believe that's the only questions that we have for this session. Well, and this marks the end of the session. So thank you very much to our presenter, Dr. Smith, and feel free to reach out to Almac Group on the platform afterwards. And thank you to all attendees for watching. Goodbye.
Thank you.